Well, hello there, and welcome back to our series of videos on the topic of inheritance. Now, in this video, we're going to look at mutations. Now, this word may throw up all kinds of images in your head. You might think of X-Men, you might think of the Avengers, the mutations that give them superpowers. But what exactly is a mutation in biological terms? That's what we're going to be looking at. What types and examples are there, and what causes mutation? So what exactly is a mutation? Well, here's our definition, which you need to know very well. So please give it a star in your notes. So a mutation is a sudden and random change, right? It's something that happens suddenly, something that happens randomly. And there are two types of mutation. So the first type of mutation is a gene mutation. So that's here. It's a sudden and random change in the structure of a gene. And the second type of mutation is a chromosomal mutation, or in chromosome number. So a chromosomal mutation will be a change in the number of chromosomes in your cells. So for humans, normally we have 46 chromosomes per body cell. And in a person with a chromosomal mutation, he would either have more chromosomes or less chromosomes than 46. So that's abnormal. It's a mutation. On a smaller scale, we have gene mutations. So for gene mutations, usually the person has a normal number of chromosomes, like 46. However, one of the genes on one of the chromosomes is mutated. It has a change in its structure. So that's a gene mutation. Now, mutations can happen in any of your cells. It can happen in your normal body cells, or it can happen in your gametes, your sperms and your eggs. Now, it's only those mutations which happen in your gametes which are heritable. Now, what that means is, if the mutation occurs in your sperm or your eggs, then it can be inherited by your offspring. Mutations that happen in your normal body cells are called somatic mutations, and these are not passed down to your offspring. So they just affect you, but they will not affect your offspring. Now let's begin with chromosomal mutations. Right? This is a sudden and random change in the number of chromosomes. So the most famous example of a chromosomal mutation would be Down syndrome, or what we call trisomy 21. It's called trisomy 21 because tri means 3, because people with Down syndrome have 3 copies of chromosome 21 instead of the normal number of 2. And people with Down syndrome have this certain characteristic facial features, and they also develop slower intellectually. So as you can see here in this diagram, this shows a karyotype of a person with Down syndrome. Now a karyotype is just a diagram showing you all the chromosomes in a particular cell. So in this person here, you can see that he has 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, one each, right? One from the mother, one from the father. However, when you look at chromosome 21, he has 3 copies. So people with Down syndrome have an extra copy of chromosome 21, and that leads to Down syndrome. Now you might be wondering, how did this person end up with an extra copy of chromosome 21? Well, it was probably due to an error in meiosis, in either the father when he was producing the sperm, or in the mother when they're producing the egg. Now I want you to imagine that there's a mother, female, and a father. And both this mother and father have the normal number of chromosome 21. So they both have 2 copies of chromosome 21. Right? Here's one, here's the other, here's one, here's the other. Now the dad forms sperm, and he forms perfectly normal sperm. Right? The 2 homologous chromosomes, right? These are homologous chromosomes. The 2 homologous chromosomes separate during meiosis and end up in different gametes. So each sperm only has 1 copy of chromosome 21. However, in this female, the pair of homologous chromosomes 21 fails to separate. It fails to separate, and so it produces an egg with an extra copy of chromosome 21, 2 copies, while the other egg is empty, doesn't have chromosome 21. So then imagine if this normal sperm fuses with this abnormal egg, it will form a zygote with 3 copies of chromosome 21. So that's something like what happens during Down syndrome. Now let's move on to a gene mutation. So remember that the gene mutation is a change in the structure of a gene. Okay, so the person has a normal number of chromosomes, but when the gene has changed in its structure, it has changed in its sequence. So an example of gene mutation is albinism, where people have absence of hair pigments, skin pigments, so they are very fair, and they're especially sensitive to sunlight. So what is the cause of albinism? Well, it's caused by a mutation in a gene called the TYR gene. And this gene is important to code for an enzyme which produces melanin. So when this gene has a change in its structure, when it becomes mutated, then this enzyme can no longer be produced and melanin can no longer be produced. Now this mutation of the TYR gene produces a recessive allele. So what that means is, in order for you to be an albino, to have this albinism, you need to have 2 copies of the recessive allele. So normally we have, all of us have 2 copies of the TYR gene. So I want you to imagine that we all have 2 copies of the TYR gene. One from our mother, one from our father. Now if one of these copies is mutated, the other copy, right, can still produce the enzyme, and so you're fine. So in people who are heterozygous, right, they're still normal. They don't have albinism. However, if both copies of your TYR gene are mutated, right, then you have 2 copies of the recessive allele, and then you'll be affected. You'll be an albino. So that's roughly how it works. Now, the other classic case of gene mutation is sickle cell anemia. So in this disease, what happens is that your red blood cells produce an abnormal form of hemoglobin. And so at low oxygen concentrations, the red blood cells become sickle-shaped. So no longer are they this nice biconcave shape, they become sickle-shaped. And that creates problems because the sickle-shaped cells will all clump together and block up your blood vessels. And they also have a lower ability to carry oxygen because they no longer have that increased surface area to volume ratio. Now, sickle cell anemia is due to a mutation in the gene that controls hemoglobin production. So again, each of us has 2 copies of this gene for hemoglobin production. One from our mother, one from our father. And the normal allele is called HbA. So let's imagine they have 2 of the normal alleles. Now the mutation causes this normal allele to become a recessive allele that produces hemoglobin S, a different form of hemoglobin which is abnormal. So in people who are homozygous dominant, which means your HbA, your 2 copies are normal, then you have a normal phenotype. You are normal. Now if you have homozygous recessive, which means that both of your copies are mutated, right, then such a person would be affected, right? So they would have sickle cell anemia. Now there is one final case where if one of your copies of HbA becomes mutated, but the other copy remains the same. So in this case, you're heterozygous, right? So this one is mutated, so it becomes HbS, right? And in this case, you are said to have something called sickle cell trait. Now, people with sickle cell trait, so the heterozygous individuals, sickle cell trait, they have a small percentage of their red blood cells which are sickle-shaped. So over here, you can take a look here. There's many normal-shaped red blood cells, but there's some which are sickle-shaped. And it turns out that even though this is sort of a disadvantage, it's a mutation, but in certain parts of the world, it turns out to be an advantage. And people with sickle cell trait, heterozygous, actually turn out to be more resistant to malaria than normal people. So malaria is a disease caused by a kind of single-celled organism. And this organism infects red blood cells. So in people with sickle-shaped red blood cells, it turns out that it actually makes them kind of resistant to this disease. And so you actually find quite a lot of people who are heterozygous in areas affected by malaria. So you'll find a lot more of the HbS allele in places affected by malaria. For example, in Africa, so you look at Africa, it's a place with a high incidence of malaria. And you can see also that the frequency of the recessive HbS allele is rather high in those areas with high amounts of malaria. So interesting, this leads to the idea of selection. Okay, so what we've just seen is that gene mutations produce new alleles, right? So from that original HbA allele, a mutation caused it to become a HbS allele, right? Now, some of these mutations can be harmful or beneficial, right? In the case of the HBS allele, it's actually a harmful allele because it causes you to have abnormal red blood cells. But in certain cases, like in places with malaria, it turns out to be beneficial. Now, when you have some people who have, when you have people with different alleles, it results in variation between individuals. And so individuals with the harmful mutation, right, they'll be eliminated over time. Over time, they will die out. While people with beneficial mutations will actually produce more and more offspring. So nature selects individuals with more favorable traits to survive and reproduce. So in the case of places with a lot of malaria, nature actually ends up selecting all those heterozygous individuals with the HBS allele to survive and reproduce. So you get a lot more of those individuals in places with a lot of malaria. So are all mutations harmful? Well, it's not the case because we've just seen that some mutations can be beneficial, just like the sickle cell trait in places with malaria. Another example is lactose tolerance in people who live in Europe. They tend to have this mutation which causes them to be more tolerant to lactose. And that turns out to be advantageous because lactose is found in milk and people in Europe drink a lot of milk. So it turns out to be advantageous in Europe to have this mutation. And some mutations as well are silent, meaning that they have no effect whatsoever on you. So it's just a change in the sequence of the gene, but it doesn't affect you. Right? So not all mutations are harmful or have harmful effect. The other thing to say is that not all mutations are recessive. Right? So far we saw that the mutations resulted in recessive alleles. So for example, when HBA mutated, it became HBS, which is recessive. However, not all mutations produce recessive alleles. Some produce dominant alleles. For example, polydactyly, extra fingers, is caused by a dominant allele, as well as dwarfism. The disease, Huntington's disease, which is one where your brain degenerates when you're about 35 years old. Now, this disease is also caused by a dominant allele. Now, I'm just going to end off by talking about what causes mutation. So one major cause of mutation is actually called spontaneous mutation, which is due to errors in DNA replication. So remember, before your cells divide through meiosis, so let's say when you're forming gametes, right? When you're forming gametes through meiosis, before meiosis, there's always DNA replication. Now, during DNA replication, what happens sometimes is that the normal gene is not replicated correctly. It's not copied correctly. Just like how when I ask you to copy out, a page of notes into a blank piece of paper, you might make some mistakes. So some mistakes are made. So in this case, the normal gene was a CTT and it was copied as wrongly into a CAT. So such copying errors result in mutations. The second source of mutation is when chromosomes fail to separate during meiosis, which I already talked about in that slide before. Now besides that, so those were the internal causes, so causes which are random, like problems in your DNA replication and meiosis. There are also causes of mutation which are external. For example, exposure to mutagens will cause an increase in rate of mutation. So some famous mutagens would be your ultraviolet light, so that's found in your sunlight, or your gamma radiation, which is found in some labs, scientific labs. So excessive exposure to things like ultraviolet light may lead to greater mutation, and some people think that it may lead to an increase in the incidence of diseases like cancer. So too much sunlight might cause skin cancer, etc. Now, another kind of mutagen are your mutagenic chemicals. So for example, tar in cigarette smoke, right? We've seen how this can actually lead to an increased risk of lung cancer. And now, of course, formaldehyde, which is a liquid used to preserve biological things like those brains in jars. And then this some of the drugs like LSD. So exposure to these chemicals might lead to more mutation. Alright, so just end off with this summary slide. Remember what the definition of a mutation is. How mutations produce new alleles, right, and hence variation. How alleles produced through mutation may not necessarily be recessive or harmful, and how mutation is caused by errors in DNA replication or by being exposed to such mutagens like radiation or chemicals.